seminar of the IBM series. And we have a pretty strong lineup of speakers till January. And the first one is Veronika Szalai from the NIST in Washington. And yeah, I read just a little bit about Veronika's previous appointments. So she did a PhD in chemistry in Yale. And then she was a research associate in a company, I guess, Anycam America, in the 90s. And then he became assistant professor in the Department of Chemistry and Biochemistry at the University of Maryland, and then associate professor, and then I suppose now she's professor, right?
Wow. It's a little different the way
I see. I see.
But that's okay.
And yeah, and so it's a pleasure to have her here. And today she's going to talk about structure and dynamics of flexibly linked multi-domain proteins using spins, scatterings, and simulations. So Veronika, the floor is yours, and you can share your screen.
I will. So thank you, Thomas. And I apologize that I can't figure out how to see all of you while I'm giving the presentation. I don't have two screens. So I'm going to have to give the presentation. And if you have questions, maybe Thomas can monitor that and somehow let me know, just chime in or something. And I wanted to say, I actually went to undergraduate and then worked in industry and then went to graduate school. So the order, I didn't get a PhD and then go to industry. I went to industry first, which was very helpful in figuring out what I wanted to do later in life. So, okay, so I'm going to share.
Oh, now you are muted. Veronika, you are muted. Okay, now you're back.
Okay, hang on. Why am I muted? What a pain. Okay. Entire screen. Okay. Share. Now can you hear me still?
Yes, yes.
Oh, good. Okay. From beginning. Let's do this. Okay. So I'm thrilled to sort of kick off the fall series for the International Virtual EPR meeting. And I'm going to tell you about some work that we've been doing over the past 2 years. And I gave a shorter version of this talk at the Rocky Mountain EPR conference back in August. And what I'm going to tell you about is how we've devised a way to measure long distances using deer on a monoclonal antibody that has been produced in mammalian cells, which means that it's not isotopically labeled or site selectively spin labeled or anything. And then we combine the outputs of those deer measurements, so the distances and the mean distances and the distance distributions, with scattering data and molecular simulations to get us more information about the structure and the dynamics of these systems. And in particular, I want to point out that the types of molecules that we're talking about, monoclonal antibodies, are just one class of flexibly linked multi-domain proteins where we don't have sufficient information about the structural ensemble that is best used to describe what the structure really is. And this is a collaborative project between the National Institute of Standards and Technology, where I work, and a couple of other principals who have joint appointments at the Institute for Bioscience and Biotechnology Research, which is a joint institute between NIST and the University of Maryland. And of course, Thomas Schmidt has been involved, and he works at the National Institutes of Health here in the United States. So I don't need to introduce monoclonal antibodies that much. They are really well known since COVID, and everyone knows that they make biopharmaceutical companies a lot of money. But I think one thing that many people forget is that they're also important ingredients or important components of any type of immunoassay measurement. So that ranges from, diagnostic tests like COVID tests or pregnancy tests, all the way to research-based immunoassays that people do. But there are some problems with being able to generate new monoclonal antibodies. So you can't always predict what's going to happen when you make changes to them. And partly that's because we don't really understand the structures of these molecules. So we tend to think of the structures as being static, but in fact, as you can see from this little movie that Christina Braganza, one of the other principals on the project, who is a molecular dynamics expert, has done, you see significant movement of these red and blue domains, which are the antigen binding fragments, otherwise known as Fab domains, of the monoclonal antibody. And so now people are trying to use AI and machine learning to try to better predict the structure and the function of these monoclonal antibodies, because they are such a big part of the bioeconomy, financially, right? So how much money they make for companies. But it is imperfect, and partly that is because, at least in our view, there are not sufficient data sets out there to train these AI systems, and data sets that contain experimental information, right? So experimental data. And so we think that we need to be able to go from a picture like this on the left, where we have these ill-defined and underdetermined ensembles. So now the Fab groups look like hair, basically, if we took a snapshot of that, of all those different structures. And we need to develop new ways to measure the interdomain orientations and distances and analyze those data so that we can get to a picture like what's on the right, where we now have discrete sets of different structures where we know what the populations of those structures are. And ideally, we would also know the kinetics of those interconversions, right? So we would not only know what the structures are, but how fast they interconvert. So as I mentioned at the very beginning, it's not just monoclonal antibodies that have this problem, where we've got multiple domains that are linked together by flexible linkers. Polyubiquitin, I know people think of ubiquitin as being, studied to death, but depending on where you have the linkages in ubiquitin systems, diubiquitin systems, that controls the dynamics, which then controls the downstream function. And the big prize that I also mentioned at the Rocky Mountain meeting is being able to figure out the dynamic structures or the structural ensembles that go into describing what happens with RNA. And so here on the right, I show a pretty recent paper from Veroni's group where they show this structural ensemble, sort of all these different structures by NMR. But you have to read the paper carefully to find out that, in fact, they developed that structural model by basically piecing together smaller structures that were determined by NMR spectroscopy. And so we would like to be able to extend the methodology that I'm going to describe to you to these types of RNA systems. And lucky for us, we've got a new postdoc who joined with one of the other principals on the group, Alex Krishayev, whose work I'll mention in the course of the talk, Karndeep Singh, who came to us from Mike Summers' lab. So he's an expert in being able to express and purify or make and purify large RNAs. And he's going to work on the protein systems as well as extending this methodology to RNAs. So I'm lucky that I work at NIST, because NIST has developed a reference material that is a monoclonal antibody. And so this reference material, you can buy it if you'd like to have access to it as well. It was developed in collaboration with an industrial partner. It is an immunoglobulin G, or IgG, class monoclonal antibody, which means that it's like all the other biologics that are used as drugs. It was expressed in a mammalian cell line. So that's really important, because it means that it has the proper glycosylation to carry out its function, right? So the glycosylation is very important in recognition events, and probably the structure and dynamics of the monoclonal antibody as well. And then it also has all of the inter and intra-chain disulfides intact. And finally, of course, because it's expressed in a mammalian cell line, it is not isotopically labeled in any way. It's stable and well documented. And so I give a reference here for the chapter in the ACS Symposium series that's on all of the physicochemical properties of the monoclonal antibody. And just an example of the sort of fluctuations in the structural ensemble that one can observe with monoclonal antibodies. These are large molecules. They're 150 kilodaltons in size. On the right, I show two scattering profiles that were collected of the NIST mAb at two different pHs. And what you can see is that they differ depending on pH, and then even the salt concentration, which are the different sort of data sets that are shown for each pH, that varies with the ionic strength, right? So if we're going to develop a way to do these long distance deer measurements, we have a couple of requirements, right? The first is we're not going to isotopically or site-specifically label the monoclonal antibody. We want to use whatever it is closest to what the pharma companies produce, right? We want it to be extendable to any IgG1 class mAb. So we're using the NIST mAb as our sort of test bed, but we want to be able to use it for any others that companies or other people would want to give us to be able to make measurements on. We want to do it in solution. So for deer, of course, we're doing frozen solutions, but the scattering measurements are all done in solution at room temperature. And finally, I mentioned again that this is large. The distances turn out to be more than 8 nanometers. And for those of you that do deer regularly, you'll know that that means I have to employ isotopic labeling of my solvent and of other sort of groups in the solution so that I can achieve those long distances, be able to measure out to those long distances. So what we've done is take advantage of bacterial proteins that have high affinity for monoclonal antibodies. And these are larger bacterial proteins. We've pulled out one domain, and we're going to use those small domains of these larger proteins as our spin label carriers. And so we have 2 different variants, constructs that we're working with right now. So the protein L domain binds to the Fabs, and the protein A, which I'll really spend most of my time talking about today, binds to the FC domain or segment of the monoclonal antibody. And so what you can see, if I can get my, let's see, my little pointer here, laser pointer. Here, if I bind the protein A's, I can get this FC dimer distance. If I bind just the protein L's, I can get the Fab-Fab distance. And of course, if both of these are bound to the monoclonal antibody, ideally, I could get the Fab-FC distances as well. Now, I want to mention that we're not the first to have thought of the idea of using a small protein to carry a spin label to the molecule of interest, the target molecule we're trying to measure. Enrique Bordignon's group has done this with a nanobody that is a system-specific protein that carries the spin tag. In their case, it was a gadolinium. And they were looking at a bacterial transporter. Of course, what we're trying to do is develop a library of these different small proteins, these SLAPs, spin-labeled affinity proteins, and we call them SLAPs because we can slap them onto any monoclonal antibody. And so these aren't system-specific, right? They're for any monoclonal antibody that's in this class. So to start, we wanted to we weren't sure that any of this was going to work at the beginning, so we needed to start with a model system. And so I spent the first few months in the laboratory figuring out how to proteolytically cleave the monoclonal antibody, the NIST mAb, to release just the FC domain of the monoclonal antibody. And then we had to pick where we were going to put the spin labels. So we need them to be minimally perturbative to the protein A, shown in yellow, orange, gold, whatever color you want to call that. We needed the spin labels to be remote from the binding interface. Of course, we don't want them to interfere with binding to the target molecule. And we wanted them to be solvent accessible, mainly because we want them to be sticking in a deuterated solvent so that we can extend the electron spin relaxation times far enough that we can collect deer traces out long enough to see the modulations, for the weak modulations that would happen with these long distances. We need many pairs of labels so that we have sufficient data to develop our structural model at the end. And again, we want the solvent and the protein, the small protein itself, to be deuterated. And I mentioned these are bacterial proteins that we are working with. So that made the expression of them in deuterated solvent with deuterated glucose possible. So we could get high levels of deuteration of these small proteins. Now, I want to point out on the right here is a little schematic showing protein A with the 4 labels positions that we used. Note that not all 4 labels are present all the time, right? So we basically step through each one of those labels when we're going to do the experiments. So we have a pair of labels bound to the FC, but we don't have 4 labels on any protein A at any given time. So the first thing I did was just confirm that the protein A's bind to FC, but don't bind to the Fabs. And so the CW-EPR spectra are very diagnostic for that. So in sort of cyan here is the individual protein A with carrying its spin tag. When you add FC to that sample, you get a spectrum that is broad and characteristic of a much larger complex being formed. And then if you add Fab to the protein A with a single spin label on it, you'll see that there's no change in the CW-EPR spectrum, indicating that the Fabs do not interact or bind to these protein A's, which we specifically selected this variant of protein A so that it wouldn't do that, but it's always good to check, of course. So I collected data, the CW data were collected on commercial instrument. All of the pulse data that I'll show you were collected on the NIST-built pulse DPR spectrometer that we published in 2018. There have been some structural, sort of architectural changes to the system since we published it. Mainly, we've got a better AWG now that is faster, has more memory, so that makes more complicated 1 and 2D experiments possible in a reasonable amount of time. We've also changed the field controller. We now use a commercial field controller rather than a home-built field controller. And for those of you who are hardware junkies, I have a picture, sort of a panorama of the innards of the bridge. So it's really a pulsed radar system. And we developed it in collaboration with Militech, which is a company that specializes in microwave engineering. And so I've taken this apart and had to fix it and troubleshoot it. I had never built an instrument before, so this has all been learning for me, but I love this instrument. It is really great at running these experiments, and it's extremely sensitive. So we are able to make these deer measurements on the FC from the NIST map. And I want to point out, however, that because of these proteins, for some reason, because these proteins are pre-deuterated, it is difficult to get the spin labeling efficiencies very high. And sometimes so here's an example of where I have 30% spin labeled A50. So I have a label in position 50 on protein A, and I was only able to get about 50% of the sample, 30% of the sample spin labeled. But what that means as a consequence is when we make our samples, because we're making complexes, we have a mixture of samples that have no signal, right? Background signal, because there's only one spin label attached, or 2 labels. And this is what's giving us the deer signal. What you can see is that it's a very low population that's giving us the deer signal. And what that means is our modulation depths are quite low, and that impedes our ability to accurately define the distribution widths. And you'll see that in the global fits that I'll show in a minute. But here's a representative deer trace for A50. And I collected biological and technical replicates of all 4 of these constructs shown here on the left. So 25, 38, 42, and 50. For K54, Thomas Schmidt provided me, thankfully, with this first mutant that he had made. So it's slightly different than our protein A's. But I only have technical replicates of that one. But I collected a lot of data using 2 different resonators on my system. I work for the Department of Commerce here in the United States, so I can't tell you the vendor names, but what I can tell you is that one vendor is blue and the other one is green. So maybe that helps you a little bit knowing which ones they are. So I fit the data globally. So either using Deer Lab, as shown here on the left, or using a single Gaussian fit to fit all the data together as a global fit. And so on the left, what you can see is that Deer Lab gives us a particular value, and I have the sigma, the RMS for it here, and it matches this mean distance matches that we get from fitting the data using a single Gaussian. And so here, what I'm showing you are the single Gaussian fits for each of the different constructs, overlaid with the molecular dynamics simulations that Christina did. And what you can see is that for almost all of the constructs, the mean distances match very, very well. In the case of A25, both the mean distance and the distribution width match quite well. And it shows that the A25 is in a rigid environment because it's so narrow. On the other hand, K54 over here, both the distance and the distribution match, and it's quite a bit broader. For A38, the mean distances match, although the molecular dynamics simulations seem to show that there are 2 populations of distances, of structures with different distances. And then for A42, although the distances don't overlap, the distribution width does. A50, as I mentioned before, we have trouble defining that distribution width accurately, but the mean distance matches very well. So in addition to collecting the deer data, I collected the echo decays to get an apparent phase memory time measurement of these different samples, different different constructs. And here are representative echo decays on the left. And I fit them with a stretched exponential to pull out that apparent phase memory time. And then Alex Grishayev, who is thinks in polar coordinates. And yes, this is a picture of Alex being happy. He's at a national park, which is one of his favorite pastimes. But what he did was he used Christina's molecular dynamics trajectory frames. He used 4,000 of those. He basically calculated the proximity of each of the nitroxide spin labels to protons nearby. And what we see is that there is a correlation between how short the phase memory time is and the proximity of protons to that spin. Based on work in the literature, Stefan Stoll's group published a nice paper where they showed that the electron-proton distance is less than about 0.4 nanometers, shouldn't be included. And Alex also weighted these data by the protein A deuteration levels. So 3 of the constructs I made were with very high deuteration levels, more than 95%. And actually, the A25 had much lower. It was 85 to 90% deuteration. So that's a really nice part of the story that I didn't present at Rocky Mountain that will be included in our publication. So it turns out I was actually excited because I thought, oh, well, now I can use that TM to tell me whether or not the Fab groups, when I use these protein A's on the full monoclonal antibody, not just FC, can I use those TMs to tell me what happens? Like, are there protons from the Fab groups that are getting close to the spin labels? It turns out that doesn't really change when you have the Fab groups. And in fact, the Deer distance measurements don't change either. So whether you have just FC, which are the solid lines, or you have the whole MAB present, the mean distances and the distributions don't change significantly. So that's good news, because now we can move on to putting groups on the proteins on the Fab groups to measure those distances without interference from the protein A's. So the distance distributions, we all know that they actually include multiple contributions. And so we've got the spin label rotamers, and then we've got protein A motion. So if protein A is moving at all, that will also affect what goes into that distance distribution. And what we're really interested, though, in understanding are these FC motions. Are there any FC motions that are there? So the first thing that Christina did was simulations of just protein A by itself. And we can rule that out as being a significant contribution. She basically characterized protein A as a rock, so it really doesn't move. The next question that one might logically have is, when you include protein A on the FC, are you dramatically changing the motions of the FC itself? And so what Christina did was a principal component analysis to show that the low frequency motions of FC with or without protein A are the same. And she did this for her molecular dynamics simulations. And I think she also did it for crystal structures, right? So they're basically the same. So what that tells us then is that the molecular dynamics that she's used, the predictions from molecular dynamics, and the experimental observables that we have agree. So now we can ask the question, are the Deer and the scattering data sufficient to uniquely define the structure and conformational dynamics of just the FC? And so this is where Alex really shines. He builds structural models. He's the one who's done all the scattering measurements and done all of the structural ensembles, creation of the structural ensembles to fit the data. And so how would we generate a motional model of FC? So we know the structure of FC from crystallography, but we don't know what the motional model is. And if you think about just generating any motional model, you have to include both translations and rotations. And when you then also include the variances on those, so you have the on-diagonal elements that are the variances, and then you also have covariances you might have to include, you can either get 27, or if you don't have to include the covariances, 12 variables that then you need to be able to use in your simulations. But if you think about this really carefully, and Alex did, you can reduce the complexity of the problem by thinking about the FC as being sort of a ball and socket. We've got an elbow here between these two different domains. They're called the CH2 and CH3 domains of the FC that you can now reduce to just having rotations. So you've basically decreased, you've gone from 27 to 9 or 12 down to 6 variables that you need to include in your model. We can also take advantage of the fact that the FC has C2 symmetry, so that further reduces the complexity. Here, it looks like I didn't actually rotate this structure by 180 degrees, but if you look carefully at this little dot here, this actually is a 180-degree rotation of the FC. And look how similar the spin label positions are when we do that. So Alex created this ball and socket motional model. He optimized what he called the interdomain pivot point, and he generated a dynamic ensemble from that. And what he got was, this is basically an overlay of 20 structures from the best fitting dynamic ensemble. And now we're going to use that dynamic ensemble to fit the experimental data that we have. And so when he uses that for to recapitulate the SACS data, you can see he used many different ensembles, many different ways of fitting these data. So he used a molecular dynamics ensemble, an ensemble generated from 40 different crystal structures, a single structure, or the dynamic ensemble. The dynamic ensemble was the one that gave the best fit to the experimental data. He then used that same dynamic ensemble to fit the deer data. And what you can see is, again, we get very good agreement, particularly in the distances for A50 and A25, and in the distribution widths for A38 and A42. He did not use the fifth data point, which was the sample that Thomas gave me, the protein A that Thomas gave me. And instead, he used that one to do a cross-validation of the dynamic ensemble, the ability of the dynamic ensemble to predict what that distance and distribution width should be. And that turns out to be very, very good. So the experimental data, we got 10.5 plus or minus 0.6. And the dynamic model prediction gave 10.3 plus or minus 0.6 nanometers. And so what that means is that we can use two low-resolution methods, DEER with our spin-labeled affinity proteins, we collect those data with the spin-labeled affinity proteins, and the small-angle X-ray scattering data to get us information about the FC dynamics. So we were sitting at a project meeting at one point. We used to have project meetings every 2 weeks. And I think it might have been Thomas, but maybe it was Thomas and Alex who came up with this. But I think at one point, one of them said, oh, well, I wonder if we can use these data to do something called rigid body docking, where we know the individual structures of protein A and FC, but let's pretend we don't know where they bind. And so, again, Alex set out to make 100 million combinations of random translations and orientations of protein A. So, protein A is floating out in space here relative to FC. And then he applied the DEER data that we have, so to restrain where that protein A could be in that 3D space. And then when you do that, you significantly reduce the number of structures that are available. So you go from like 100 million to something like 10,000. I can't remember the exact number. And what that does is it brings you to a collection of structures that are within 7 to 1 nanometers of the correct structure, which we're using as the crystal structure of the FC protein A complex. If you apply now a SACS filter where you also restrain it based on the SACS, which tells you about the electron density of the complex, you can refine that even further and get down to under about 2.5 nanometers. And finally, in order to get as close as we can to the crystal structure, we applied a small computational filter, which was an interfacial surface area filter. Basically, the idea is that if you're really making a complex, you need to have some sort of minimal binding interface that defines that. And we used an interface area of 2 square nanometers and 200 square angstroms for those of you that still think in angstroms. And so what that looks like in 3-dimensional space is this. So when you apply the DEER filter, you get basically a barrel of protein A's around the FC. When you then apply the SACS filter, you can narrow down where the protein A is, but because SACS is not sensitive to the orientation of protein A, you can't completely define that structure, the complex, the structure of the complex. And it's also important to remember that protein A is very small compared to the FC, so it doesn't contribute significant amount of electron density to the SACS scattering profiles. But once we apply the interfacial surface area, we are now able to really home in on where the protein A is bound to FC and what that looks like in a larger structure format where we show protein A in licorice, and the different colors tell you how close we are to the RMSD of the crystals, to the crystal structure. we then get a model that tells us where protein A binds to FC. So in summary, what we're able to do is combine two low resolution methods along with molecular simulations, including molecular dynamics simulations, to not only tell us about binding of two species, right? We can also get information about the dynamics of FC. And these dynamics haven't been shown before for FC, but we know that FC, this is the part of a monoclonal antibody that binds to receptors in vivo and different cellular receptors. And it's never been clear why that happens. And we think that understanding these dynamics could help us get a handle on that. So these dynamics are likely important in controlling receptor binding in vivo. So I want to thank Cristina Berganzo for all of her work on the molecular dynamics simulations. The monoclonal antibody is huge. It takes quite an effort to put the whole thing together and be able to run these long and complex molecular dynamic simulations. Alex, of course, has done all of the scattering measurements and the simulations, and he's really been just a wonderful colleague. I love talking to him and learning how he thinks about building models of molecular structures and thinking about how to simulate them in the best possible way. Thomas Schmidt, I didn't mention him enough. He really helped me with the deer. I had never done these types of measurements before. I'm a bioinorganic chemist, so I was raised doing CWEPR spectroscopy of biometalloproteins. Basically, I had never done a lot of pulsed work. And he was very generous. He not only shared a sample with me so I could get started and see if there's proof of principle, I could get it to work, but he also shared all of his Python codes for me to be able to do the fitting and the analysis. And he just generally is a very energetic and supportive person in terms of the project. And finally, Karndeep, who's come onto the project with his expertise in RNA and NMR, who's going to be working on both the protein parts of the project and on extending it to RNA systems. Because I don't do Python, I've had some help from some folks at IBBR who are much more experienced at it. And Gail even helped me set up using the IBBR server so that I could basically do calculations or fitting there. There are other folks at IBBR who also have been helpful. Subrata is doing biomolecular NMR, Jay Kuhn. Some of you might know him. He's a solid state NMR spectroscopist. And then, of course, I couldn't have done any of the biomolecular labeling without the support of the Biomolecular Labeling Laboratory, which is at IBBR. NIST doesn't have facilities to do that. So I've really relied on them. And Zvi Kelman, who's the director of that laboratory, has a joint appointment. Rachel and Kinlan are both expert molecular biologists, and Kinlan's actually a structural biologist as well, who helped me a lot. And finally, I'd like to thank Brad Odell for coining the word SLAP. We were trying to think of how to think about these proteins, and now we're able to say that. So with that, I'm going to end my presentation because I can't see any of you if I don't. So now I can see everybody.
All right. Thank you, Veronika, for this very interesting seminar. We really appreciate it. And now I open the floor for questions. I invite anyone who has a question to raise the hand and maybe unmute himself or herself. So yeah, we have a question from Andreas, the first one I saw. Probably.
Yeah.
Hi, Andreas. Go ahead.
Hi. So thanks. It was a cool talk. Maybe there was a detail and maybe I misunderstood because there were constantly people coming in and asking stuff. But you said your labeling efficiency was low because you used deuterated protein. Is that I don't see really the connection between deuteration and labeling efficiency. So is that something?
Well, I can't say for sure that it's all the deuteration. Here's what I know. When I make a protonated protein and I make the deuterated protein and I side by side spin label them, the one that is deuterated is more badly behaved. It tends to aggregate and precipitate and do things that I don't like. So that cuts into my labeling efficiency, right? So that's what I've observed. Whether there's a reason that deuteration should do that or whether it's the construct we've chosen, I can't say for sure, but I have run the side-by-side sort of, race, parallel, spin labeling. And that's what I've observed.
Okay. Yeah, thank you. Interesting. Interesting.
Yeah,
Didn't
I don't know
this.
why. And I've asked a lot of people. And I talked to Christina about it, the molecular dynamics person. She's like, well, we don't worry about deuteration. When we do our molecular dynamics simulations, we don't worry about that. And I'm thinking, but it's got to be different, right? I mean, the protein's behaving so differently, and I don't know why. So.
Okay. Thanks.
All right, we have the next question from Thomas.
Hey, Veronika, thank you so much. I know I'm actually involved in that. Hey, Veronika,
ask me a question. Go ahead.
Fine. Look, I just want to. I figure like maybe can you make a comment that because actually the antibody they were actually glycosylated, right?
Yes.
But so that's very important because normally proteins are not glycosylated if we express it with E. coli or anything. So this is actually one of the biggest parts of the equation, really, here, that it's still glycosylated. But I'm curious, does the glycosylation, does the level of glycosylation, does it vary between the different antibodies? Would that have an effect?
The level of glycosylation or the type of glycosylation?
Both. Actually,
Yeah,
the type. Let's talk about the type.
I'm going to have to be honest and say I don't know. Christina would know the answer. Maybe Alex knows the answer, but certainly I've seen that different antibodies have different glycosylation. Whether the levels are different. I mean, part of what you do when you're developing a drug, right, is to show that you've got the same drug molecule, like that the distribution of glycosylation in the vial that you're giving to somebody is the same every time. So if you're asking whether it's 100% glycosylated, well, I guess at some level it's got to be, because otherwise you couldn't get it through like FDA drug approvals, right? So you have to show that the glycosylation level, whatever that is the same across every batch that you do, right? Now, whether all the sugars are the same, I don't remember. I'd have to look that up. Yeah, I don't remember. Cool. Maybe Alex can chime in. He's here. I saw him. He's here. Maybe he could chime in.
There is a statistical distribution.
Okay.
Of course, statistics.
That's what I would have assumed, right? Is you get some distribution, but it has to be the same every time. That's for sure when you make a drug.
Sure. Cool. Thanks.
Okay, we have a question in the chat from Anna. I read it for you. It's I wonder what is the significance of this structure in terms of ESR measurements. I'm not sure if I understand the question. Maybe Anna can comment on that.
Yeah, I'm trying to understand.
I think if I have understood, she would like just to know, me too, I do have this question. So if this procedures that you have used for your own system can be extended to other systems, to use this protocol to look at the older systems, or each time you have to use specific proteins to tag them on your system under study?
Well, okay, so we've developed a library of proteins that are specific for monoclonal antibodies. So there's tons of IgGs out there. These proteins will work for any of those IgGs. So in that way, no, you can't take these protein A's and stick them on, I don't know, some iron transporter in bacteria and have it work. You would have to find a small protein that you want to use as your spin label carrier, right? That is specific for your system. But ideally, what you'd like to do is get something that binds to a class of, say, ABC transporters or something, right? Where it's not a nanobody, where a nanobody is raised to have a recognition site that's only for that particular protein partner, right? Target. So you don't, you could use nanobodies, but the problem is that then you would be only able to use them as far as I understand, for that particular, construct that you're trying to measure something on. You couldn't say use it on, I don't know, one GPCR and then use it on a different GPCR, right? Because the loops would be different and the recognition site would be different. So what you'd ideally want is something like, let's say a G protein, right? A small G protein that binds to lots of different GPCRs, and you could use that, right? So it's not extendable, like you could use R proteins for anything, but it is extendable in terms of being for a specific class of proteins, if that makes sense. Does that help?
Yeah, thank you. For me, thank you. I don't, but yeah, that's fine. Thank you.
Does that answer Anna's question?
I think so.
She's
I
still
hope
here?
so.
She's still here?
I don't think she's here anymore. I don't
Okay.
know. I don't see her.
Okay.
Yeah. Are there any other questions from the
Veronika,
audience?
can I ask a question?
Yeah, sure.
Yeah, it's a great talk. I liked it. Okay. My question is, see, as you said, this binding protein, you cannot use it for any other proteins. It's just for IgG. So now what I'm thinking, this IgG binds to Fc gamma receptors. Can we use a small domain of the receptor so that in that case, that you can spin label it, attach with the antibodies, then you can use it for all the antibodies, like, for all the IgG subtypes, everything can bind to the receptor, so that we can find out the distribution of this structure that help us, how they bind to the antigens. So that
you're way ahead. I mean, we've definitely gone down that path, but I think it's hard to think of a way that well, maybe I'm misunderstanding, but you want to spin label the receptor protein?
Yes, so if we can take a small domain of the receptor,
yes,
where the Fc binds to.
Whichever receptor you want, yeah.
Yes, then we can study this structural distribution.
Yes.
That help us, when the antigen binds, how the structural distribution will be.
So you're absolutely right. And actually, I'll tell you that back in 2015, when I started thinking about this project, so I couldn't work on this because I didn't know anybody who did monoclonal antibody work. So I reached out to a company and they were going to work with me, but things at companies change rapidly. So by the time we got started, they changed their mind. So I didn't get to work on it until recently with Alex and Christina, because they had also been thinking about this type of project. But that's originally what I wanted to do was, actually, what I want to do is include non-native amino acids in the monoclonal antibody and spin label with nitroxides and gadoliniums, so that I had gadoliniums in the Fabs and nitroxides in the FCs, right?
Yeah.
But the whole point was that they wanted to know, what is the structural distribution of the Fabs when it's bound to antigen, when it's also bound to receptor? That's what they wanted to know. So you're exactly right.
Yeah.
In that is the direction, that is definitely a worthwhile direction to pursue. It's a lot of work. And right now, at least, I didn't mention this, but like all of the experimental work was pretty much done by me, right? So I'm the equivalent of a full professor, and I'm doing all this work in the lab, essentially, right?
Yeah.
So it's fun. I love being in the lab. Don't get me wrong, but it's tough to problem solve every single thing and get complicated projects like what you're describing really off the ground and do them carefully. So
the one more thing is, the one more question is, recently, ADC, antibody drug conjugates.
Yep.
You can attach a spin label to somewhere in the drug. So when they they design the ADC, when you attach the spin label, because spin label is a small molecule, like some nitroxide, but of course nowadays the TAM radical can also be used. So we can attach a small spin label to the drug domain, the drug.
Yeah.
Then how the drug binds to the antibody when they make the ADC. So we can study the structural distribution. That will be very helpful in the now when people make the ADC, the antibody drug conjugates for any treatment. That will be very useful, how that ADC can bind to Fc gamma receptors.
Yeah, so the thing is, there's so many things we've talked about being able to do. It's basically a resource shortage, right? So we can think of lots of ideas, lots of really good ideas, like the ones that you're talking about. But when you have like one person in the lab, it's hard to pursue all that. I don't have a big group. It's me and maybe one other postdoc. It's not a huge number of people. So, but yeah, these are all wonderful ideas. Thank you.
Thank you. Great talk.
Thank you.
All right. Are there any more questions from the audience? In the meantime, I claim the host. Okay, if not, I think we can all thank Veronika again for a great talk. It was really a pleasure to have her here. And for everyone, let me share my screen. Yep. All right, so then this is the lineup of the speakers that we have, and our next seminar is on October 9th, and I hope to see everybody of you there. And here we conclude this seminar, and have a wonderful rest of your week. I have to go for a hurricane.
Thank you, Thomas.
Okay.
Thanks,
Yeah, all right.
Bye.
Thank you.
Thank
Bye-bye.
you. Bye-bye. Hey, Thomas, are you still there?